Hello, everyone. Thank you for giving me the opportunity to share some of our results today with you. So as the title says, it's about the role of alpha-synuclein aggregation in multiple system atrophy. So alpha-synuclein is here one of the key players in my talk. And so alpha-synuclein is actually a very abundant protein in the brain. It's a presynaptic protein, a very small protein of about 140 amino acids long. And so it's been known for a long time, since it's one of the major components in Lewy bodies and Lewy neurites and Parkinson's disease, but it's also present in so-called glial cytoplasmic inclusions and multiple system atrophy. I'll come back to that. It's also been linked genetically to a rare form of autosomal dominant Parkinson's disease, where it's been shown that point mutations or gene duplication or triplication can cause these rare forms of Parkinson's disease. And so as you see here, this protein is normally present in a soluble form, and under certain circumstances, it can aggregate into insoluble amyloid fibrils. So by intermediate steps where you have small oligomers, then amyloid fibrils, and these are the fibrils, the aggregates that you find in Lewy bodies or in glial cytoplasmic inclusions. So one of the questions is, of course, what determines this process of aggregation and how can a cell protect itself from it? What has also been shown more recently is that the protein can even travel from one neuron to another neuron. So it's kind of transmitted from neuron to neurons. And so this reminds actually of a behavior that was known before from prions. So one of the questions is that alpha-synuclein might also behave as a prion-like protein. So we're talking here about alpha-synuclein. And so alpha-synuclein is actually involved in different diseases that are termed synucleinopathies. And so Parkinson's disease is the most frequent one, but you also have dementia with Lewy body and multiple system atrophy, of course. And so if you look at the symptoms of these diseases, they are actually quite different. So there's some symptoms here displayed for Parkinson's disease, which are quite different in a sense from multiple system atrophy, and also different from dementia with Lewy bodies. If you look at the pathology in the brain, also here, we know in Parkinson's disease and in dementia with Lewy body that alpha-synuclein aggregates in neurons, and so it's called Lewy bodies, but in multiple system atrophy, it aggregates actually in a different type of cells in oligodendrocytes, and there it's called GCIs, or glial cytoplasmic inclusions. So one of the mysteries is how this one protein can be causing or involved in these different diseases. And so what researchers like my group do is to try to model these diseases in rodents. So we try to model both Parkinson's disease or multiple system atrophy in rodent models. And so we, of course, not the only ones who do this, and you have different ways to do this. So one way is to make transgenic models. So what people have done is to overexpress alpha-synuclein in neurons to model Parkinson's disease, or in oligodendrocytes to model multiple system atrophy. Another way is to use viral vectors, so where you actually inject viral vectors and overexpress alpha-synuclein in the brain locally of animal models. And also here you can do this in neurons, trying to model Parkinson's disease, or in oligodendrocytes, trying to model multiple system atrophy. And a third way is that researchers have found that if you inject fibrils, what we call here recombinant preformed fibrils, so aggregates in the brain of animals or in other organs, that you can kind of mimic the transmission, the prion-like transmission of this protein. So one of the expertises of my lab is to use viral vectors to model alpha-synuclein aggregation and neurotoxicity. And very briefly, we started to do this a long time ago, mostly in the context of Parkinson's disease, so where you have a viral vector that you inject in the brain of, or in the substantia nigra of rats or mice. You induce overexpression of this protein, and this gives rise to a number of phenotypic changes like dopaminergic cell death, and you can monitor this with dopamine transporter micropet, where you can see that you lose the signal over time unilaterally, because we usually inject on one side of the brain. Or you can do this also by histology, where you can count dopaminergic cell bodies or the dopaminergic terminals, and you see a time-dependent decrease, or the time-dependent loss of the cell bodies and the terminals. And we also have alpha-synuclein aggregates that are induced. So you have kind of Lewy body-like inclusions induced in the brain of these animals. And what we also have, of course, that was too fast, is we also can follow the animals by behavior. And so there's different tests that have been developed to look at the use of the front paws. And so if there is a deficit on one side, you will see that the animal will use the contralateral paw less over time. Or you also have this rotarot, where the animals have to stay on a rotating wheel. And so if they are impaired in motor behavior, they will fall off and they will not be able to stay on this rotarot. And so this is also something that you can use to monitor the behavior, the motor behavior of the animals. So again, we want to use these animal models to try to understand a bit better how alpha-synuclein is involved in the different diseases. And so one of the concepts that we have worked on is the concept of strain. So this concept of strain comes from prions, from prion diseases. And to explain you a little bit what this means, a strain means that you have, for example, what you have here, you have your soluble unfolded alpha-synuclein that can aggregate into these fibrils. And so the shape of the fibrils can be different. So it's a bit like you can imagine with Lego blocks with the same units, you can make different kind of constructions, three-dimensional constructions, or you can also kind of compare it with different types of pasta where you have different shapes, like more spaghetti type or more linguine type. They're made from the same component, but they have a different shape. And so this can be called strains. And so we started to collaborate with Ronald Melky, who is a biophysicist from Paris. And so he found that in vitro we could generate from alpha-synuclein different kind of strains. So he called them type 1 or type 2, and here he called them fibrils or ribbons. And he could show that in vitro and in cell culture, these different polymers behaved quite different. So they had different characteristics in terms of structure, resistance to proteolytic degradation. They also showed different levels of toxicity in cells and different seeding and propagation properties. So we tried to investigate the properties of these different strains in the rodent brain. And so Wouter Peelaerts, who was then a PhD student in my lab, he started this project. And so what he did actually was to inject these different strains in the substantia nigra of rats. So you have here the fibrils, the ribbons. As a control, we used oligomers of alpha-synuclein. And so we injected them in the substantia nigra of rats alone, or in certain cases in combination with our alpha-synuclein viral vectors, which are displayed here. And the idea is to display more soluble alpha-synuclein to probably enhance the phenotype or to accelerate the phenotype. So what he did was to look at Lewy body-like formation if you inject these strains. And so indeed, what we saw that injection of fibrils or ribbons induced alpha-synuclein aggregates in dopaminergic neurons. So you can see it here by staining for phosphorylated alpha-synuclein together with TH as a dopaminergic neuron marker. And we also found Lewy-like neurites, so filamentous inclusions. And so what was quite remarkable is that if you compared the ribbons and the fibrils, the shape was quite different. So the ribbons induced more these filamented inclusions compared to the fibrils, where you had more different round inclusions. Another thing we noticed, which was quite remarkable, if we injected ribbons in combination with our viral vector, so to supply more soluble alpha-synuclein, We also found inclusions not only in neurons, but also in oligodendrocytes, as you can see here by the co-staining with the OLIG2 marker. And so these inclusions resembled kind of GCI, so glial cytoplasmic inclusion that you find in multiple system atrophy. So this was a first link that this might be related to more multiple system atrophy instead of Parkinson's disease. We also looked at the neurotoxicity of these strains, and this is actually a table that summarizes really a lot of experiments, but I don't have time to go into detail. And so we compared the dopaminergic cell loss, the dopaminergic terminal loss, and the motor impairment in these animals. And so the conclusion of this is that the fibrils were actually the strain that caused most neurotoxicity on all these different parameters, more than ribbons, and oligomers were actually causing almost no toxicity. We also looked at crossing of the blood-brain barrier. So we injected these strains fluorescently labeled in the bloodstream intravenously. And so we looked if they could reach the central nervous system. We did this by imaging, but also by histology. And indeed, we could show that they can cross the blood-brain barrier and that they form inclusions in the spinal cord and in the brain. And so what you can see here, they also cause a kind of microglial reactivity, and the fibrils were actually most causing this deposition and this microglial reactivity. So to conclude this first part, we could show that injection of recombinant strains in rat brain induces Lewy-like pathology with a different phenotype. So the ribbons cause more a filamentous phenotype. And we also could show that in the presence of ribbons, we also find some inclusions in oligodendrocytes reminiscent of GCIs. And fibrils are the most toxic species in vivo. So the conclusion of this is that this might indicate that indeed difference between alpha-synuclein aggregates in multiple system atrophy and Parkinson's disease or dementia with Lewy body could be due to different strains present in these diseases. Now, to investigate this bit further, we wanted to go more to patient-derived material. And so what we did is we obtained material from the brain of different patients, some MSA, PD, and DLB patients. This was actually in collaboration with Steve Gentleman from UCL, who have this brain bank. And so this material was then sent to Ronald Melke in Paris, where he isolated, so he made homogenates of this brain, and he isolated actually the alpha-synuclein from these brain samples in a technique that is called PMCA, so where he can really specifically amplify the alpha-synuclein in a strain-specific manner. And then we kind of characterize these strains in cell culture and in the in rat brain. So the results of this were that, first of all, we looked in vitro. This was actually done in Ronald Melke's lab. So he could indeed show that the strains that were amplified from this patient material formed fibrils, but the fibrils looked different under an electron microscope. And so the conclusion was that the fibrils from PD and MSA patients looked more similar to each other than, for example, the fibrils derived from DLB. Also in some other tests like proteolytic degradation or recognition by conformational antibodies, it also looked at there were differences between the strains, where again the PD and MSA than the DLBs. Was quite a large experiment of viral vectors in the substantia nigra. And so we followed the animals over 5 months by behavior analysis. And then at the end, we looked at the brains and we characterized the brains for different parameters. So what we have here per group of disease, we had 4 to 6 patients, and per patient, we had 3 animals. So it was quite a large group of experiments. So I will just highlight important results because there's a lot of different results and they have been published. So we looked at motor behavior. So each time we have either the homogenates or the PMCA purified, amplified fibrils. And then we have a group where you have the alpha-synuclein homogenates alone or in combination with viral vectors. And so in terms of behavior, what we saw the most striking results were here the combination of the homogenates with the AAV vectors, where we could see that the MSA-derived material caused the most striking behavioral impairment compared to the other groups. We also looked at then the brains, and so we looked at dopaminergic cell loss, again for the different groups, and we looked at the loss of the again, I will highlight the results. What we could see that the derived material caused most dopaminergic cell loss. And also, if you look at the nerve terminals, also here, the MSA-derived material caused most dopaminergic nerve terminal loss compared to the other groups. Then we also looked at alpha-synuclein pathology. So what we did with stained for phosphorylated alpha-synuclein, which is a marker for pathological inclusions. And also here, you see that the MSA homogenates induced much more phospho-alpha-synuclein positive inclusions. We found them in dopaminergic neurons or in striatal DARP32 medium spinal neurons. We did not find inclusions actually in oligodendrocytes. So it seemed like in our conditions, we had only neuronal inclusions. Then we looked also at transmission or spreading over the different brain regions. And so what you can see here is, so we looked at different brain regions, as you can see here. And so we pseudo-colored the different locations. So where red is most extensive alpha-synuclein pathology, yellow is a bit less, and green is also less. So it gives a visual representation of the pathology that we found. And so what was striking is that in both groups, so both the homogenates or the PMCA-derived fibrils, the MSA group shows the most extensive spreading and pathology compared to the other ones. And strikingly, the DLB showed actually less alpha-synuclein pathology, and PD was a bit in between. We also looked at immune response. So to do this, we stained actually for different markers. So we have IBA1 as a microglial marker, then MHC2 for antigen presentation and infiltration of different types of T cells. And also here you see the summary of the results with the number of pluses to give a more strong reaction. And also here you see that the MSA group shows the most pronounced immune response, both in the homogenates and PMCA fractions. So the conclusion of this study was actually that in almost all of our parameters, the MSA group showed the most extensive phenotype, more severe phenotype, which can be reminiscent also if you look at the disease. MSA is actually much more aggressive disease than Parkinson's disease or dementia with Lewy bodies. So the conclusion of this part is that we could show that if we derive alpha-synuclein strain from patients, that we also can that they show strains with different structural traits that are characteristic for each disease, that the MSA strains are more similar to the PD-derived strain compared to the DLB strains, and that they induce the most pronounced phenotype and immune response in vivo. So then the last part that I would like to discuss is actually a most recent part. So it's actually just published, it's just in press. So what we wanted to do here is we wanted to go to a more MSA-like model. So we chose an Msa transgenic model. This is a mouse model that has been developed and characterized quite well, that expresses alpha-synuclein under an oligodendrocyte-specific promoter, the PLP promoter. And so we wanted to combine this model, which shows already quite an interesting phenotype, but not always a very severe phenotype. And so we wondered what would happen if we would inject these animals with the recombinant strains that I expressed previously. So the fibrils, the ribbons, and then the BSA. So we injected them here in the striatum, and then we analyzed the animal at different time points, 3, 6, and 9 months post-injection by motor testing and then by analysis of the pathology in the brain. So first of all, we looked at motor deficits. And so here we have actually as a control, we injected BSA as a control for a protein. And then we had the two different ribbons and fibrils alpha-synuclein strains. And so what we saw that over time, the animals, and so there's two tests here. There's a pole test where you let the animal actually climb onto a pole and then descend again. And so the speed of doing this gives an idea of the motor behavior of the animal. And so we saw that mostly the animals with in the fibrillar conditions showed a time-dependent deficit in this test. And also in the cylinder test, we saw that the fibrillar group actually showed a decrease over time in this motor tests. Then we looked, of course, also in the brain. And so we looked at the dopaminergic neurons in substantia nigra, but we also looked at the neurons in the striatum and the DARP32 positive cells in the striatum. And so what we saw here, so we have always a contralateral site which is not injected and an ipsilateral site. So the contralateral is actually the control. And so we saw here that there was a time-dependent decrease in dopaminergic neurons, also in DARP32 neurons in the striatum, or in neurons, total neuronal cell numbers in the striatum, in the fibrillar condition, and so less with the ribbons and not with the BSA condition. Then another striking thing is if we looked at the brain, so what you see here is the different groups, actually representative sections. And so if we look at the striatal volume or the ventricle volume, it was quite striking that in the fibrillar condition, there was an enlargement in the volume of the ventricles and a decrease in the striatal volume. And so this was quantified also here. So you see that there's actually kind of shrinkage of the striatal volume and an increase in the ventricular volume in the fibrillar condition. And then we wanted to look a bit more specifically. So we did a staining for myelin to see if there was some demyelination. And this is with a dye that is called fluoromyelin. And as you can see here in the fibrillar condition, you see a clear demyelination in the corpus callosum, but also in the striosomes, which is not present in the other two conditions. So this is the quantification here. We see a quite severe demyelination in the fibrillar condition. Then what we also wanted to do is to look at the different alpha-synuclein assemblies. And so what we use is these kind of new probes. These are called luminescent conjugated oligothiophene or LCO probes. And so what they do, they bind to fibrillar structures in a specific way. And so to first characterize them, we looked at brain of human control tissue or brain of MSA patients. And so we stained them either with phospho-alpha-synuclein, which is the marker for pathological alpha-synuclein, or with these probes. And you can see the quantification here. So in MSA brain, we see clearly phospho-alpha-synuclein inclusions, which was known for these GCIs. But you also see specific binding of this probe in the MSA and not in the control condition. So we knew that, or could show that these probes can detect these GCIs in the human brain tissue, a bit similar for the phospho-alpha-synuclein and the probe signal. So then we wanted to look, of course, in our animal models. And so what we saw here, if you look at phosphorylated alpha-synuclein, that the injection of the ribbons and the fibrils gives an increase in phospho-alpha-synuclein inclusions compared to the BSA. But if we stain them with this probe, this LCO probe, you see that in the BSA condition, there's actually almost no signal, but it's clearly induced with injection of ribbons or fibrils. And this is actually the quantification of the number of, or the number of phospho-alpha-synuclein and LCO probe positive cells. And you see here that it's clearly present in these animal models, in the ribbons and the fibril condition. It's about half or double stained, so it's not all the inclusion that are stained. And if you compare this to MSA brain of patients, you can see that there's actually a complete colocalization of the phospho-alpha-synuclein and the LCO probe, which is less in the animal condition. And so we can also further characterize this. And so what you can do with this probe is look at the emission spectrum. So you can do a spectral analysis and see, and this spectral analysis will give some information on the specific structure of the strains in these sections. And so what we could do this for the different cell types. So we can do this in oligodendrocytes, in neurons, in microglia, and astrocytes. So you get a spectrum information on the structure of the inclusions in these different cell types. And what was quite interesting to see is that in the BSA condition, you have a quite large spectrum, which means that there's different conformations present. But if you inject with the strains, you see that the spectrum becomes much more narrow and much more specific and different for the ribbons and the fibrils. And if you compare this to the spectrum in MSA brain, you see that the ribbons are more similar to the spectrum in MSA patients compared to the fibrils. What was also striking that if you look in different cell types, like in neurons or in microglia, that the spectra are also different for the different strains, while, for example, in astrocytes, we could not see any difference. So it means that the ribbons and the fibrils, they induce inclusions in the different cell types that are different from cell type to cell type. Then we also looked at other phenotypic changes. And so, for example, if you look at astrocytes, we could show that the injection of these ribbons and the fibrils induces astrocytic activation compared to BSA in the brain of these transgenic animals. And then if you look at the colocalization with phosphorylated alpha-synuclein, we see that the ribbons induce much more inclusions or much more specific inclusions in astrocytes compared to the fibrils. So the number of double positive cells is much larger in the ribbon condition compared to the fibril condition. Then we also looked at microglia. And so what we saw that the ribbons and the fibrils, they induce indeed a microglial activation. So with no difference no difference between the two groups compared to the BSA. But if we look then a bit more in detail at the CD68 marker, which is a kind of antigen presentation marker, we see that the ribbons induce more CD68 expression compared to ribbons and much less in the BSA condition. Or we can also look at MHC2 marker, and there also here we see that this can be used as a marker of reactive microglia. We also see that the fibrils induce a more pronounced increase compared to ribbons, and certainly compared to BSA, where there's almost no reactivity present. Then we also wondered whether there was only local immune reaction in terms of microglia, or whether they were also infiltrating peripheral immune cells. And so we can do this by looking at different markers. So we looked at CD45 positive cells, and we could see that indeed, especially in the ribbon condition, but actually also a bit in the fibril condition, but also a bit in the ribbon condition, that there is infiltration of CD45 positive cells. And then if you do a staining of IBA1, which is negative for TMM119, so TMM119 is a marker for resident microglia. So with these, you would stain actually the infiltrating macrophages. We can see that there's a clear induction of infiltrated macrophages in the fibril condition, also to a lesser extent in the ribbon condition, but certainly much less in the BSA condition. And then finally, we also looked with a marker for T cells, with the CD3 positive cells. And also here we see that there's clearly infiltration of T cells in the fibrotic condition, less in the Rubin condition, and almost none in the BSA condition. So to conclude here, so what we could show in this study is that the injection of recombinant alpha-synuclein strains in an Msa transgenic mouse model modifies and accelerates the disease phenotype in a strain-specific manner. So it triggers clear oligodendrocyte-specific pathology and neuronal pathology with inflammation markers. And so we think that the combination of these strains in the transgenic animal generates a unique animal model that more closely mimics the human MSA condition than the transgenic animal model alone. And so to conclude, we think that we have some indication now that MSA might be the consequence of an interaction between specific alpha-synuclein strains and the specific cellular environment. And so we hope that this will give some more information on the specific disease mechanism in multiple system atrophy. And so I would like to thank here by, or to close here by thanking several people who have contributed. So here, these are the people in my lab who have mainly contributed to the experimental work. So Anke van der Perren, Wouter Pelaerts, Geraldine Gelders, and Teresa Torre Muruzabal, who have contributed to most of the findings I have shown. But of course, other people in my group have also contributed. And then we have some funders who provide the funding to do the research, and then some important collaborators that have also contributed to our studies. And I would like to thank you for listening to my results.